Okay, so this is the part third of biotechnology. If you have not watched the, so you should watch that first. So now we are going with the recognition side. Now what I'm making here, so this is the my gene of interest. Okay, so now what in last video we have studied about the phosphate, these all groups. I'm writing here first one G A T T C, and here it is. Here it would be opposite of this, so G would be here, A T T C, and here of course it would be same like that, G A T T C, and here it is C T T A G. What is it? It's the fifth prime, and here's third prime. Of course, if here's the 3, so now it would be here. There's third prime, and here's the fifth prime. Now don't think like what I'm making here is all. I've taught this all in the previous video. You should check that first. So now what will happen here, if I'm making plasmid is like this kind of a structure. Okay, so now what are plasmids? If you're taking an bacteria first, here's the bacteria, okay, like bacterial cell, or we can say, okay, this is its DNA, and like we are having more structure in the bacteria, like these are known as plasmid. What are now plasmid? Like these are double like structure, the round one and this round two. What would this do? Like plasmid would transfer gene from one to another, like from DNA. This is also known as non-essential DNA. These plasmid are also known as non-essential DNA. Plasmid, these all are plasmid. These are non-essential DNA. Even this is the DNA, but these are the non-essential DNA which has been like transferring the gene from here to here. It will also move in like one bacteria. We are having a lot of like 150, varies with the bacteria size, cell size, and this is the double like structure as I told you. So now what will happen here, this one is plasmid. We have taken one plasmid and showing it to you. So now what is this? This is a plasmid, and as I told you, the here is my gene of interest first. Okay, so what will I do? So like here is my gene of interest, so I am writing here first one G A T T C, and here of course the same G A T T C. These are the as I told you, these are written here, so I've brought these first. And now what will happen, this is a crazy, like we can say, acto 1. Okay, this would be coming here and will be showing the gene of interest. So now what will happen, like it will show the gene of interest. Here we can see the difference. This is starting from this, and this is starting from the end, like there's no bond, and here's also no bond. So this means now what will happen, maybe like I make one, like you can get confused, like I have showed the one bond, so let me make little bit big. Okay, so this is that plasmid. Okay, so now here's G A T T C, and here's C T T A G. Okay, so now G, here we can see it's little bit far, here's no any bond. Okay, and here it's also, here's the last one, this means it is also not having any bond. First one is this. If we make another, let me move like this. Okay, so here's another G A T T C, and here's the second one C T T A G. So now what will happen here, you can see it is also not having bond G1, and here C is also having last bond like this. So here G separated, here also G separated, and here G, here G. Okay, now what will happen, if I wanna separate this all, here's that. So now what will happen, like these are separated. Now what would be the condition, here only G is remaining, the unparticipated, like they are having bond, like this would be having bond with this is having this, like these would be straighted. I have made a little bit far from this, here would be no any bond. Okay, so this means the one who is not having bond, I will write here G, and the bond they are participating. And here in the end what will happen, even this is also like, and like here these are making bond, so here I will write CTTA. Okay, so this means G here is making bond with this one, okay, but not with this one. So like here GCT, like here G is not making bond, and here the remaining are not making bond. Okay, so now what would be in the second one? So yeah, I will write the same. Here we can see, like, let me show you. Here G is not making bond, so first we'll write the G1. Second, here what here first one, like here it's making bond, this one with this, like these all are making bond, but still the one is remaining. Which one is, like here AATTC is making bond, but here only G is making bond. Like here the others are remaining, they are not making bond. And now what that bond is known as, like they are having hydrogen bonding with them so that they can attach easily. But here still, so like these are done, but the bonding, hydrogen bonding is with them, but still here if you want to add more bond, like the same CTTA, and here if you want to add G, the same. So there is one enzyme, we can say what that enzyme is. Let me write enzyme, that is DNA ligase. Okay, DNA ligase. Okay, what will that do? Like that will attach, it's a, like it work as a glue, even we can say. It will attach other bonds together. So again these would be attached. So this was the biotechnology, like this part third, as I told you. So now what's the work of restriction enzyme here? Restriction enzyme, now what is that? Of course we are talking here about the restriction recognition sites. So what's the work of restriction site? First thing, it is prokaryotics, like these will match with the prokaryotic, sorry. And second one, it is like bacteria, it's save bacteria from viruses to DNA. Of course, if it would be in the bacteria, this is the even we can say our, what does our skeleton do in our body? It's help in the protection. So likewise, it is enzyme which help and save or like inside work. Like here bacteria, it would also damage DNA. If you're talking about here bacteria, so it can even damage DNA. So that's the reason it's a restriction enzyme which helps in, like, not, it's help in protection, like we can even say that. And after like cutting DNA, if we cut any DNA, so there we can even find the restriction enzyme. Okay, so this was all for today.